now I give the floor to our next speaker, Dirk Redecker, who will, with him, we will enter a different world of the symbiosis between fungi and roots. So you have the floor.
So good morning, everybody. Thank you very much for inviting me and giving me the opportunity to present this topic and also to present our collection in the context. So the collection I'm talking about here is the International Bank for the Glomeromycota, which is hosted at INRAE Dijon. Good. Okay, so I was about to explain what kind of fungi we are working with, because these are rather unusual microbes. We are working with Arbuscular mycorrhizal fungi, which are in the phylum Glomeromycota. So Mycorrhiza, as you well know, is a mutualistic symbiosis between plants and fungi. And the fungus acts as a kind of extended root system for the plant and absorbs mineral nutrients and phosphate. And in turn, the plant will provide carbon compounds such as sugar, but there's also examples for lipids. There are several types of mycorrhiza. Two of them are shown here. So this is ectomycorrhiza, and I will concentrate today on Arbuscular mycorrhiza. And Arbuscular mycorrhiza, the great majority of land plants associates with fungi from the phylum Glomeromycota. On the plant side, these are mainly herbaceous plants, but also quite a lot of tropical trees and some of the most ancient plant lineages. On the fungal side, this is a relatively small group of fungi. So the Glomeromycota contain only about 300 described species. Recently, it has been shown that there's also fungi from another phylum involved in an association with which resembles very much Arbuscular mycorrhiza. These are the Mycorrhiza, but this is not really well studied at the moment. Arbuscular mycorrhiza is in particular known to be involved in the nutrient absorption of many crop plants. So instead of naming all the plants involved, which is the majority, it is more useful to cite the non-hosts, which are shown here in examples of this. These are the Brassicaceae with cabbage and mustard, and other plants like spinach and rutabaga. In the photo, you can see what happens if you leave a dependent host plant without a mycorrhizal partner. So on the left-hand side, the soil was sterilized, and many plants don't grow very well. On the right-hand side, the soil was not sterilized. There were mycorrhizal fungi present, and you see that the growth is much improved. Some effects of most mycorrhiza associations actually are listed here. So this is in particular an increase of the absorbing surface by the mycelium, which you can see in the background. The mycelium will transport mineral nutrients from the soil, and this is in particular important for those nutrients which are not very mobile in the soil, such as phosphate. If you have a complex system of multiple plants and fungi, a redistribution of nutrients in a common mycorrhizal network is possible. This involves nutrients like phosphate and nitrate, but also carbon. Apart from the nutrient effects, it is well known that mycorrhiza increases pathogen resistance under certain circumstances by inducing resistance. And an overall effect of mycorrhiza on plant soil ecosystems is an improvement of soil structure by forming soil aggregates. So culturing Glomeromycota is not very easy. These are not fungi that you can just grow on an artificial growth medium. These are obligate biotrophs. That means they have always be to cultivated with a living plant host. In most cases, this is done as it is shown on the left-hand side here in open pot cultures. This has the disadvantage that the spores that are harvested from these cultures contain a lot of other microorganisms. Some fungi are available in what is called in vitro culture. These are cultures on an artificial growth medium, sterile, with transformed roots. And as you can see very nicely in the photo, these are spores that are forming on the roots that are colonized. The problem with this is that this is only available for very few species. Host specificity in the cultivated isolates, at least, is very low. So you don't have to think about which fungus to use for which plants, at least in principle. However, in semi-natural systems, host preferences have been detected. As I said, the collection that I'm directing in Dijon is the International Bank for the Glomeromycota. In this collection of Arbuscular mycorrhizal fungi, AMF, we have over 40 isolates, and this slide presents the morphology of the spores of some of those fungi. The fungi are mostly cultivated in pot culture, as I explained before, on their host plants, and these are put into growth cabinets, which provide the necessary light for the growth of the plants. Some of them, as I said, are also available in vitro. So overall, we have 43 isolates in 30 species. On their plant hosts, 6 of these are available also in vitro on transformed carrot roots. So we do this in parallel. The origins of our isolates are very diverse. So they are mostly from Europe, but there's not really a real focus on France. So there's only 3 French isolates in our collection. We have North America, South America, Oceania, and some from Asia. In particular, I would like to point out that 11 of our isolates are X-type. So this means these are the standard isolates for each species. 43 isolates doesn't seem to be a lot if you say it like this, but you have to keep in mind that these isolates are cultivated all the time. So we don't put anything in the fridge and freeze it. All the isolates are living. So to use these mycorrhizal fungi, which, as I said before, are improving mineral nutrition and may add to pathogen resistance, there are 2 different approaches basically possible. The first approach is to maintain and support existing indigenous species. So that means in the field site where there's a community of AMF, you have to adjust the conditions to maintain and maybe to increase abundance and diversity of the indigenous fungi. So among these conditions that you can change or you can modify are tillage, the level of pesticides, fertilization, and in principle, you have to avoid rotations with non-hosts, but because this will not increase the mycorrhizal population in the field. Another strategy would be useful mostly when there's only a very weak indigenous community, and that would be to add fungal propagules by inoculation. Inoculation can be useful, but also challenging because the inoculated fungi have to compete with the indigenous population. In many case studies on mycorrhizal growth stimulation, the researchers have simply not verified this. So I will present a study where we have done this in several minutes. So in general, inoculation seems to be easier and better established for plants that are pre-grown, for example, are grown in sterile potting substrate in the greenhouse, and then they are outplanted to the field. So it has been shown that plants that have been pre-grown in the greenhouse or even under sterile conditions profit a lot from mycorrhizal colonization when they're outplanted and their survival rate is increased. One aspect that has to be taken into account also, which is not directly mycorrhizal technology, is more like the circumstances that can be modified, is the influence of plant breeding. So it is well known that plant cultivars differ strongly in their mycorrhizal response. So it really matters which variety of wheat you are using. And there have also been data that breeding may, in fact, decrease the mycorrhizal dependency. So it would be good to establish cultivars that interact well with the symbionts, which then would facilitate the two other approaches. So for the first approach to improve the conditions for the indigenous mycorrhizal communities, you have to understand what actually happens when agriculture is present and what the different kinds of land uses do to the mycorrhizal community. In this study that has been published in 2003, the colleagues studied a range of field sites, and there was a gradient of land use from a very, let's say, natural permanent grassland towards maize monocropping with several intermediates. And what was emerging was that the number of fungal species in these fields decreases in the direction of the intensive agriculture. And this has been, in principle, being confirmed in other studies. So obviously, agricultural practice has a very negative effect on the diversity and also on the abundance of arbuscular mycorrhizal fungi in the field. So one of the factors that are acting in agriculture is obviously tillage. So the irregular destruction of the mycelial network in the soil, in the intensively tilled soils, obviously represents a strong stress factor for fungi. Apparently, certain species of AMF are more able to deal with this stress factor than others, and these may be the ones that propagate quickly and form a lot of spores. In this study here, we compared the community structure of AMF in different plots in a long-term field site in Switzerland, where 3 different methods of tillage were performed. These 3 treatments were conventional tillage, chisel, where the soil is just cut and not turned, and a treatment without any tillage. The different colors represent the different molecular species we detected, and within each treatment, we analyzed 4 different plots. Overall, the effect of tillage on the communities was evident. There are different colors in the different treatments, and there are some species that simply disappear during in the intensive tillage treatments, and some only survive in the no-till treatment. But why should we worry about AMF diversity? So in a classical experiment, Marcel van der Heijden confronted a seed mixture in a greenhouse experiment with different levels of AMF diversity. And the result was that with a higher diversity of inoculum, a higher diversity of plants above ground could establish. Moreover, the productivity of the system increased with inoculum diversity. The effects were quite striking, as you can see from this photo. This is a seed mixture on the left that was confronted with a high diversity of mycorrhizal fungi, and on the right-hand side, there was no inoculum added. And you see that on the right-hand side, there are mainly grasses that establish. However, what does this mean for agriculture? In an agricultural field, there's only one host plant. Does it really matter if there's a high diversity of AM fungi present, or is a single species of AMF that remains in the field that is efficient enough for the purposes of yield increase? So in this study from New Caledonia, the authors inoculated plants in an ultramafic soil, that is a soil which is very poor in phosphate and rich in heavy metals. And they inoculated the different treatments with the indigenous plant Metrosideros laurifolia, with the single species or with a mix of species. And the overall result of this was that the species mixture was the most efficient. That is, the plant growth was best when there was a high diversity of species present. They conducted a similar experiment with sorghum, which is not a native plant, obviously, to New Caledonia, and the results were in the same direction, but less straightforward. So another aspect to be taken into account is that a large part of Amf diversity may be found on the intraspecific level, and inter-isolate variation, that is intra-species variation, has to be taken into account. So in this study from 2004, Munkvold and co-workers in Denmark showed that the yield, that the plant growth was very different with different isolates of the fungal species Funelliformis mossae. So these are the bars here on the left-hand side. So you can see that the shoot pea uptake, for example, was differing between the Funelliformis mossae isolates. In another study, Burstler et al. Showed that there are possible ecotypes of the very widespread species Rhizophagus irregularis. There was a genetically defined group of isolates that were only found in grasslands and never in cultured soils. So there might be a large intraspecific diversity in Arbuscular mycorrhizal fungi, which is difficult to address because these isolates are difficult to distinguish. As I mentioned before, it is rarely done that inoculation success is actually controlled in growth experiments with Arbuscular mycorrhizal fungi. That is, it has to be verified whether the inoculated fungi really colonized the roots. So in numerous studies that has not been done, I present you one where we did it. This inoculum tracing may be difficult because in many cases the inoculant strains are from the same species as the species that are anyway found in most agricultural fields. So you cannot distinguish them. You need markers on the isolate level. So in this experiment, Phalaris arundinacea, a grass, was used to revegetate a mine spoil in the Czech Republic. You can see the field here. And in this experiment, the researchers were interested in the question whether mycorrhizal inoculation could improve the establishment of this grass. And in this experiment, we also tracked the inoculum. That is, we verified that the inoculum actually propagated and established in the roots. We used mitochondrial markers to characterize the inoculum strains. And in fact, in the beginning, we were not very amused because in the inoculum, there were two different genotypes, haplotype A and B. It turned out that haplotype B did not play a big role in the experiment, so the effect of this was not very severe. So what was done was using these mitochondrial markers to determine the frequency of samples that were colonized by haplotype A in the inoculated plots, which are shown in dark green, and in the control plots, which are shown here in light green. And this was done throughout the experiment over almost 3 years. So it turned out that in the beginning, after inoculation, the haplotype A was not present in the plot, which was a good thing for the experiment. And it did establish after some while and increased in frequency for about 1.5 years. And after that, it seemed to fade away a little, but we did not look long enough to really look if it would disappear. In the non-inoculated plots, the haplotype established much later and never at the same frequency as in the inoculated plots. So what we can say here, it was significantly more frequent in inoculated plants. I also talked about in vitro cultures, this kind of culture where you can produce very pure fungal inoculum. And I already mentioned that this is only for very, very few species. Basically, this is possible for Rhizophagus isolates. This has the advantage, as I said, of pure inoculum, but of course, the technological effort is higher than in pot cultures, especially for large-scale production. There's also the dilemma that most in vitro inoculum that is produced worldwide is actually from a single fungal strain from Canada, which is Rhizophagus irregularis DOM 197, 198, because this is the one that grows best in the in vitro cultures. So if you buy spores that are produced in vitro, the chance is very high that you have this isolate. As I already mentioned before, a plant breeding could also be a door opener for mycorrhizal technology, because when we have the wrong plant cultivars, mycorrhizal technology will not have the same impact. It has been known for quite a while that plant cultivars are known to differ in mycorrhizal growth response and root colonization. And there was the hypothesis that breeding and domestication actually had a negative effect on mycorrhizal growth response, because during breeding, this normally was not a criterion for choosing a cultivar. So this may well have a negative impact on mycorrhizal impact in the field. And this is why researchers looked at changing the situation if mycorrhizal colonization could be a criterion in breeding, but the results were not so encouraging. I present you here this study from Liza et al. In 2016, where they did a genome-wide association study. And what you see basically in the diagram is the probability that a given locus, a given gene, has something to do with the trait that is indicated. So in the lower panel, we have single tiller biomass with a significant threshold, and you see that there are many points above the threshold which correspond to genes that have something to do with single tiller biomass. You see, however, in A, that for mycorrhizal root colonization, none of these points reaches the threshold, which means that heritability of this trait is very low, and it may be distributed onto different loci. Oops. So, in summary, I would like to list the points why mycorrhizal collections can do something for sustainable agriculture and for agricultural research. So mycorrhizal collections, as ours, can be resource centers for research inoculum production. They can be resources for develop better culturing methods, improve detection and identification, study functional diversity and the interactions with plant cultivars, and they will provide well-defined inoculum isolates for industrial partners for inoculum production. In these collections, in my view, interest-specific diversity of what I call the common colonizers, which are the species that you find almost everywhere, which are listed here, has to be represented, as well as the taxonomic and functional diversity outside of this, that is species that you don't find commonly in agricultural fields and which also may have an important ecological role. So at the end, I would like to thank the collection staff, in particular Nathalie Kosela, who is a technician working in the collection. And I would like to thank also the INRAE agents that are at the moment installing our new automatic watering system. And I would like to thank you for your attention. And I'm looking forward to your questions.
Thank you very much, Dirk, and thank you for this informative talk. For me, as my colleagues know, and now I am proud to be able to pronounce glomeromycota without any mistake, because it is a new world for me. So we have a question from Muriel, so you can invite her to?
Je peux pas.
No? Ah.
Il y a un problème technique avec elle.
Ah. So, but Muriel, you can...
Elle est dans la discussion, dans la question.
Or maybe we can read your question if we cannot invite you to join the scene.
I can see the question.
Oh, yeah, you can see the question directly. Good.
Okay, so the question was, all mix did not lead to better plant diversity. What was the mix specificity in mix 5 and 6? Okay, so in these mixes, there was always as a main component Esophagus neocaledonicus, which was always present. And there were different species in there. I think there was one additional species in mixture 6, but I think I can look it up. I have the mixture here. Mixture 5 and 6. 5 had 5 species and 6 had 6 species. That was the difference. So there was one Acalospora species in addition. So actually number 6 had a higher diversity than number 5. That is right. So there's not an easy correlation between species number and effect here. It's much more complex.
Okay, so we have a question in the room. Thierry, maybe you come close to...
Hi, Dirk. This is Thierry speaking. Do you hear me?
That's difficult.
Okay, so come here. No, finally, that's this system.
Hi, Dirk. So I have 2 questions. The first one is regarding the diversity between the isolates. Do you plan to isolate more, to include more isolates in your collection, in your Glomeromycota collection, to increase the number of isolates in your collection? And the second one, do you have one or several explanations of the low heritability of the mycorrhization? So thank you.
Okay, thank you for the question. So we are quite limited in space in the collection. So this is why we only accept new isolates when there's really scientific interest. But generally, yes, I think it would be a good idea to accept new isolates from these species to better be able to study the isolate, so to say, cultivar diversity in AM fungi on the intra-species level. But we cannot accept isolates at a large scale because simply there's not enough space there. We're already quite limited. So this all the isolates that we accepted recently were type strains, which otherwise would have died, and there has to be scientific interest. But generally, yes, good idea. So heritability, that's a difficult question. I'm not sure if I can say much about that. My best hypothesis for this is that there are too many different genes working together. And this is why it's difficult to have a selection on this. So there's a large range of species involved in signaling during the in the beginning of the symbiosis and during, and maybe that's too complex to just simply breed on this.
But Dirk, if I may just add, if you have a low heritability, it means that at that stage, maybe, environmental components are more important. I don't know, water content of the soil or chemical composition of the soil. It doesn't mean you don't have a genetic control, but that the environmental variance is much larger than the genetic variance.
Okay.
So you can still have genes,
okay,
but you have a higher importance of environmental factors.
Okay, thanks. This was what I said was an hypothesis that the authors cited in their conclusions. So, but as I said, I'm not an expert on plant genetics, so I'm looking forward to interactions in this field.
Yeah. But that's the fact that it is a ratio. So you can have a low value because you have a low genetic component, or because you have a low value of heritability because you have an enormous environmental component.
Okay.
So that's always the problem with ratios.
Okay.
Is there other question in the... Anne-Françoise, another question from the room?
So, good morning. It's about one of your first slides where you were listing the challenges to set up a collection of Glomeromycota. And in one of the points was that the spores were often associated with other microorganisms. And I was wondering if it was only a problem of mixing several species of this fungus, or if it was really different, other completely different microorganisms. And if you know if there are some collaboration in Natura between these microorganisms to establish an interaction with the plant.
Yeah, that's a very interesting question. So basically, this is not what I'm talking about here is not contamination with another AMF species, but there are bacteria, saprophytes, and even pathogens associated with the spores. And there have been studies that shown that they are sitting within the spores. And there's some very interesting studies about bacteria that are obligately associated with some of those isolates. They have been described as taxonomically described already, and their genome has been analyzed, and they are associated with certain strains and certain species, and influence also the biology of the fungus. So these bacteria increase, for example, the germination. The strains that have been cured from the bacteria germinate less. So there is definitely some interactions with these microorganisms. And in the case of bacteria, it has been shown that they can be obligatorily associated, and there's other studies that have been shown that some bacterial lineages have been associated with certain AMF species for a very long time in the scale of millions of years. But in everyday use, this is just the problem is that you find saprophytes even within the spores, the saprophytic fungi, bacteria, and others. So this it's actually microbes zoo, a spore like this, and this has really hampered genetic analysis.
Thank you very much, Dirk. So we have the last questions and then the break. So the question is from Gwendal Restou.
Yeah, thank you for this very nice, interesting speech. I just have a question, in fact, which follow up the question about heritability, but you mentioned that selections for cultivars could have a side effect by diminishing the mycorrhizal growth rate. So did you quantify this negative correlation, or did you plan to do it?
Well, I have not worked on this myself. This is from literature. And this is an idea that has been around in mycorrhizal research for quite a while. It was brought up in the 90s, and it is a little strange that there was no really clear follow-up on this. So I'm not sure if you can quantify this correlation really in the way you mentioned.
Okay, because it seems very similar to question we can have also with in animal breeding with microbial diversity and these kind of things, kind of side effects of selection. So I'm very interested in.
Okay, that's interesting to know. Yeah. Okay.
Okay. Thanks a lot.
So thank you very much, Dirk, and thank you for the previous speakers.